All right, recording begins. It is November 14th, and this is ESM 105. Thank you all for making it. Okay, first thing we're going to do is take a quick look at the syllabus, right? See where we are in the course. Right here in week 12. So we're starting pesticides, poisons, and pollution. We're making some progress on that one. You've got this week, next week, and then a break. Don't forget about quiz 5, which is going to be due on November 16th, and which is on the week 10 through 11 Ecological Economics and Human Populations PowerPoint. It's 27 multiple choice questions, so it's pretty long. But I gave you 120 minutes to do it, and I recommend right here, just get all your references together, have like a snack and a cup of coffee and all that stuff ready to go. Take a little 10-minute break if you need one. You got 2 hours to do it. So, just take your time with it. The answers should all be, you should be able to find out all of them in the PowerPoint. Okay. Otherwise, don't forget about weekly discussions. There's another one open today. And don't forget about the annotated bibliography, which, it'd be great to be making some progress on. Otherwise, I think we're ready to start. Are there any questions? Okay. Cool. Okay, we are in our poison section right now. We're in the section of the PowerPoint where we define a bunch of terms, really. So let's see, I think we got to the antibiotic resistance slide, which if you're watching the recording is slide 16. All right, and I was talking to you, it's a standard issue line. I'm sure you've gotten this before, maybe from a doctor, maybe from a bio class, somewhere, about how, make sure you complete your whole regimen of antibiotics, right? Because you don't want to allow antibiotic resistance to evolve. Let me full screen this thing. If you think about it from an ecological perspective, when you put an antibiotic in an environment that bacteria are trying to grow in, you are introducing a selective pressure, okay, where individuals that have genotypes that make them resistant to antibiotics are selected for. So based on our rules of evolution, right, or natural selection, okay, you're going to see an increase in the frequency of that gene, whatever gene or whatever allele is letting the species resist the bacterium. And there's lots of ways that species can have antibiotic resistance. Some of them exude enzymes that break the product down, the antibiotic down. Some of them actually sequester the product inside of themselves or bind it with other compounds to make it non-active, which is going to be interesting later as we start to talk about bioremediation. Okay, but I'm just trying to plant some seeds there. Anyway, key way in which antibiotic resistance becomes more prevalent. You put the antibiotic in, it kills all the individuals that aren't resistant to the antibiotic, but if you don't take the antibiotic long enough, the antibiotic-resistant individuals repopulate, and now you've got a whole population of resistant individuals. And then you've got this additional interesting aspect present in bacteria of horizontal gene transfer, where they can transfer genetic material from one individual to another individual. Sometimes they don't even have to be exactly the same species, but they can form this little conjugation tube between individuals and pass a little piece of DNA between, which allows them to, say, resist an antibiotic. All right? And plasmids are these extra chromosomal pieces of DNA, so these little small circles of DNA that have important genes on them. They reproduce independently of the main chromosomal DNA, so they can be passed between individuals, and we use them in genetic engineering all of the time as ways to put genes into bacteria. So it's pretty interesting stuff, this plasmid transformation and the use of plasmids. It's all important. Anyways, if you want to do plasmid transformation, just take Bio 105 with me. It's like a fun class. It's a lot of work. It's a very stressful class from the student perspective. but we do a lot of fun stuff in those labs. Anyways, let's talk about healthcare, environmental toxicology, and then define some more terms. So pretty intuitive statements coming at you here. The heaviest burden of illness is borne by people who can't afford adequate health care or afford to live in a healthy environment, in an environment free of pathogens, in an environment free of poisons, you know. I'm going to avoid using the phrase toxins as much as I can because as you will see later, that's a commonly misused phrase. It does have a definite biological meaning, but reporters and articles that you read even, they'll use the phrase toxins when they don't mean toxins. So we're gonna say poisons, which is more accurate. Okay, awesome. Not a lot of resources for health. Health goes down. Fair enough correlation right there. All right, solid. And with global exchange of biological material and global exchange of individuals, putting more funding into developing countries where pathogens aren't being dealt with in the modern correct way could actually have global benefits, right? Because you're fighting against the prevalence of antibiotic resistance, for example. Cool. Toxicology, okay? Toxicology is a field of study. When something ends in ology, it's a field of study, right? Whether it's biology, ecology, pharmacology, virology, all these different things, okay? Toxicology is the study of poisons, okay? Not toxins. And I'm sticking on that because, as you know, I'm using a textbook as a primary source for the structure of this course to kind of give me a framework. But I'll tell you right now, I keep finding more and more wrong things in this 5th edition textbook that's being assigned in college classes. It's wild, all right? They define toxicology as the study of toxins, and it's not at all true, okay? Toxicology is the study of poisons, and I'll explain why in just a moment on this slide right here, okay? So, what are poisons, all right? Poisons, general term, a substance that kills, injures, or impairs an organism. You can tell I wrote this. Impairs an organism through chemical action. So, a poison's poisonous. You know what the word poisonous means from interacting in the world on an intuitive level. It impairs the function of an organism. Maybe it impairs the function so much that it kills the thing. Whereas a toxin is specifically biologically produced. So we can generate toxins inside of our bodies. Fungi can generate toxins. Bacteria can generate toxins. But you don't generate a toxin in a lab. You generate a poison in a lab if you're trying to build a pesticide or something. If it's not biologically derived, it's not a toxin. Okay? So you can go out from this class with another opportunity to correct people, which makes us loved by all of those around us. Right? Next time somebody says a toxin, you can be like, I think you mean a poison? If you just give me a minute, and then as they walk away, you can be like, wait, stop, please, I'll stop, I promise you. All right? Anyways, toxicology studies poisons. Really, it studies things that are toxic, okay? What are toxic things? They are known poisons, okay? That damage, kill cells, kill tissues. So, if something is affecting organisms via its chemical action, it's toxic, it's poisonous, it's a toxin. Any of those words could apply. But there are other hazards that also reduce dailies. Remember our DALY, which was a standard issue measure. Oh my gosh, where'd it go? A very standard issue measure for the impact of a hazard on humans, right? That is disability adjusted life years, which is really usually it's in 1,000 people, how many years of quality of life are lost due to this environmental hazard, whether it's toxic or not, okay? I know it's a weird metric to keep harping on, but it's used in so many of these reports on global health because it's more, you can consider it as more representative than just saying reduction in lifespan or reduction in, the average age at death. It's actually a reduction in quality of life. And who defines it? That's another question. Dailies could be estimated differently by different groups, but that's always the case with any of these metrics. You can measure it differently, you can present it differently. Okay, awesome. So there are things that are hazardous that are not toxic. Toxic things are poisons. Poisons act via chemical action. They break something down. They impair the functioning of a metabolic pathway that would otherwise function normally. Okay, they're poisons. Toxins, biologically produced. Something like ricin is a biologically produced poison, so it's a toxin. Okay. Allergens are specifically substances that activate your immune system. So they aren't necessarily working via chemical action. It's not a poison. Though it might feel like it. So if somebody walks up to me and throws a handful of pine pollen in my face, I'll feel like I was poisoned, like I was exposed to a poison or a toxin. Okay? But what's actually happening is my immune system is being stimulated. It's misidentifying those pollen grains as invading organisms. This is an oversimplification, but it's good enough, And therefore, your body sort of starts to do these standard things that it does when it's being attacked by a pathogen. Think about what your body does in response to a mosquito bite. A mosquito bites you, and it swells up, and it gets itchy. Why? Well, it swells up to try to prevent the movement of things out of that area where you received the bite, all right? It gets itchy, it gets irritated because of the differences in blood flow that are happening there, all right? Think about what happens when you get a bunch of pollen in your face, all right? Your eyes start to water, your nose starts to run, you start to cough to get the air out of your body. Your body is inducing large-scale changes to make you eject things out of your body. That's what it's doing. Because being allergic to something is usually considered, it's weird to say a malfunction, but it's considered as like a malfunction of your immune system. Your body's identifying that pollen grain like it was a worm trying to crawl into your brain, all right? If it's a worm trying to crawl up into my brain, I want to be blowing my nose a whole lot. But I don't necessarily need to when a non-toxic, non-poisonous pollen grain gets lodged in there. So allergens interact with your immune system, and they elicit an unpleasant response, because when our immune system is firing off, it's making you do things that disrupt normal life. Please.
Are venoms necessarily biologically derived?
Yes, venoms fall under toxins, so biologically derived poisons. Yeah, awesome. And you can have toxins that are generated inside of your own body as well, right? A brilliant example doesn't spring to mind, so I'm just gonna move past it. Cool. Okay, more terminology. Let's just keep throwing it at you here. We've got antigens, all right? So allergens are usually also active as antigens, but antigens specifically, your white blood cells activate them, and you get antibodies developed to identify those antigens again later. So this can be this is a function of like a correctly working immune system. Something gets introduced into your body, it's got antigens on it, that generates an I don't want to say biotic response, but it activates an immune response through your white blood cells. But so it's really easy to think like, well, why is that different than an allergen? Okay, but it's more specific. And if you mobilize the white blood cells and get production of antibodies, then you end up being better able to deal with the next incoming round of this organism. Some things screw with your immune system, and that's no good. Make it too active, make it unable to respond to invaders. Yeah, that's a bad situation, okay? Some things are endocrine disruptors. Your endocrine system is usually discussed as the system of hormones moving through your body. So you've got these special tissues that make up these special organs or these special glands, and you got like your gland system, all right? It's making all of these hormones that go through your bloodstream, and they elicit large-scale responses in your body. Like you've heard of stress hormones, right, as one example. So if something disrupts your endocrine system, it can lead to large-scale changes in the organism and how it responds to stimuli. Or it can just change how you feel, put you in a constant state of panic, which your body needs to be able to get you to do. When you're in danger, it needs to be able to release hormones to say, like, make you more vigilant, make you more focused, stress you out. But you don't want to constantly be up at that level, okay? So the stress hormones are an oversimplified example because there are a ton of important hormones in your body that could be disrupted. But if something's identified as an endocrine disruptor, all right, well then that is something that's disrupting your normal hormone functions. Environmental estrogens were a big thing back when I was a student, So like 2 years ago, right? I'm 25. Very good, everybody's buying it, we're fine. BPAs, right? Everybody was super concerned about these, and now plastic bottles still say BPA-free on them. Coffee cups will still say BPA-free lining, right? BPA was an environmental estrogen mimic, okay? And so it looked like estrogen, and it could possibly behave like estrogen. The studies on this were all a little bit vague, of like how much your body would actually respond to the BPAs, but there was enough research out there to make people concerned. And then folks jump on it like they tend to do with one specific environmental issue, and BPA gets removed from all these products, okay? But some sources of BPA, if you're actually concerned about it, get entirely ignored. So BPA is produced in a lot of plastics as a byproduct of plastic production. And what's made of plastic are our toilet seats. But nobody thinks about what's coming into your body when you sit on a toilet seat. So nobody ever regulates BPA concentrations in toilet seats. But I don't know if you spend as much time as me on the damn toilet, like I'm probably getting exposed to a lot of BPA. Because I got Instagram reels to look at, I got TikToks to record, I got all these things to do, yes.
I've heard that another silent killer for BPAs are thermal paper. So like
Oh, it's huge.
Yeah.
It's
because it's so thin, your skin just.
Yeah, and it's thermal paper, so it gets heated.
Yeah.
So it's like you've got this hot little sheet of BPA-rich paper, and you get handed it, and you go like, "Cool, thanks." You crumple it up in your hands like this, and you like, I eat them because I'm in a zero waste lifestyle right now. Yeah.
Same.
Good. So yeah, BPAs. People used to be way more concerned. I just asked my Bio 105 class, and there's like 70 people in there. I was like, who's still like talking? Is anybody still hearing about BPAs? And like 2 people raised their hand. I was surprised, but it could be the effect of different majors. In this class for the recording, about 80 people, all right? Who here is still hearing about BPAs? Oh, wow, a lot of people. Great. Now, please, quiet down. There's too many of you. Stop rushing the front of the class to talk about BPAs. Anyway, different majors have different things on their radar. I just love the toilet seat example, and it is relevant to our discussion about how we're getting exposed to poisonous substances, okay? Because it's really easy to have a blind spot. You think about the water bottle because you're drinking the stuff that sits in it, but you don't think about the thing that you're sitting upon. Anyway. Cool. We got I think we're into our last 2 slides of categories. We've got neurotoxins. Neurotoxins, okay, are poisons, not necessarily toxins, which is annoying, okay? But they specifically affect nerve cells. So they affect your nervous system. They're really bad because if your nervous system starts to fire off incorrectly, everything goes haywire really quickly, okay? Then you can even just injure yourself or, become incapacitated and you're driving a car, all these things. Okay, no good. These are some different categories and they act in different ways. We're not going to really worry a ton about this because it's not a course where we're talking even about nerve cell structures or the nervous system. But just know that there are multiple categories of things that affect nerve cells, and they all fall under the umbrella term of neurotoxins. The idea here is that when you're going out and doing conservation work, you'll understand the terminology when someone says, oh, well, this is a neurotoxic chemical, or like, oh, this is a teratogen. All right, you want to know what these terms mean. Mutagens, what do? Mutagens induce mutations. They generate mutations, so they change your genetic sequence in some way. If your genetic sequence gets changed by mutagens, that can be pretty bad. You can get cancer from it, you can get tumors from it. Sometimes your system can recover from exposure to a mutagen and correct the shift in DNA sequence, okay? But sometimes these mutagens can be so intense and harmful that the cell just needs to be destroyed, okay? If that doesn't work, sometimes the cell becomes tumor tissue, where it's no longer regulated, it just starts to divide and produce this blob of undifferentiated tissue that doesn't have any of the regulations or the functions that the initial cell was intended to have. Next, we've got teratogens, okay? And these are specifically chemicals that affect embryonic growth and development. So if you're exposed to these while you're in the womb, okay? So we've got alcohol can operate as a teratogen, okay? Leading to fetal alcohol syndrome. There are a lot of other chemicals that can act that way. So the question with these is often, right, can it pass through the filter of the placenta? If the chemical can't pass through the filter of the placenta, then it might be a mutagen, it might be a poison, it might be a toxin, it might be an antigen, it might be an endocrine disruptor. But if it can't pass that filter, if it can't get to the developing embryo, then sometimes it just doesn't have any effect on the developing child. So teratogens are focused on that development stage. And carcinogens cause cancer. Okay, you could have something that was mutagenic and carcinogenic and teratogenic, which you'll see come up in our example of Love Canal, which was a garbage dump that they built a school on top of and a whole neighborhood on top of, a toxic waste dump actually, and then everybody started to get real sick. You know, we'll talk about it. I've got some additional details that'll be interesting. Okay, cool. Yes, please. That's a great point. I forget where I got that source from, or what was cited for that source. Because when we saw our daily tables, cancer wasn't included in those, but that's depending on that's actually a really good point to emphasize right here. If we're looking at the disability adjusted life years, cancer doesn't even, really fall onto here. But it does cause a lot of death, all right? So it's, it's interesting in that way, like how many years of quality of life were reduced versus how many people die to something are different data points. So while I don't have that answer for you rocked and ready to go, yeah. It's really cool to see a good example of that. Like, depending on what data I present, I could show that, say, smoking doesn't actually harm people that much, Because, yeah, you get cancer, but it doesn't even rank on the list of top 10 losses of DALYs, which is how I measure how harmful something is in the environment. Yeah. Good stuff. All right, diet. So we've talked about the poisons, we've talked about that, and now we talk about how diet can lead to cardiovascular disease, which is one of the major reductions in dailies in our table, or cardiovascular diseases. US, you get into an area where food availability is taken care of and it's readily available, and then the food starts to get more and more processed. We've got a different handling of food in the United States. than in maybe other countries. And I am not prepared to go into an in-depth discussion of that, how like, well, why don't other countries that have readily available food not have the same obesity problem? I don't know. I'm technically obese. I love it. I get down hills so quick. It's not an appropriate thing to joke about, but you know, I'm very self-conscious about my weight. And yeah, part of it's definitely due to my diet. Why am I talking about this from a personal perspective? Let's totally take it out of this, okay? It's another cause of reduced quality of life, okay? Reduced quality of life in overweight individuals due to cardiovascular disease or consistent chronic pain. These are all things to think about if we're using our consistent metric of DALYs, right? Okay, cool. We're starting to get into the stuff that I'm a little more excited about. All the definitions are just, it just gets so dry and repetitive, you know? So, we're going to talk about how, oh man, this is no good. This is a slide that should not say toxins. It should say if I could spell the damn poisons, it should say poisons. I thought I edited any slides that looked like that, okay? Because I got pissed when I was like looking at this material. Like everything says toxins. That's wrong. Stop teaching students incorrect things. And then I just tried to do it to you. Okay. Things move through the environment, right? Poisons move through the environment. Some poisons are more stable than other poisons. Some can be degraded by heat, some are degraded by life. We measure how stable a chemical is based on its half-life, which is going to come up in just a second. But whether or not something can move through the environment, it also is dependent on the solubility of that poison. So I don't think, there's nobody here from my Bio 105, right? No, okay, cool. I see so many faces, you know. This is one of my favorite things to discuss in Biology 105, which I teach every semester, which is hydrophobicity and hydrophilicity and polar versus nonpolar molecules. You'll be amazed at the dividends that you will receive if you invest in understanding hydrophobic, hydrophilic, polar, and nonpolar. Okay, those aren't in the right order, but let me just explain them real quickly. Water's a polar molecule. Remember way back in like week 1, I talked to you about water and pH and stuff for some reason. I had different ideas back then about how much I could cover in 50 minutes in this room. But water's a polar molecule. So it's got a positive end and it's got a negative end. All right? We've got a partially negative charge. We've got a partially positive charge. Ooh. So a solution of water, it's like a big old bucket of magnets. And if you put something that is magnetic into the bucket of magnets, the magnets are all going to surround it, okay? And that sort of pulls that thing into solution and holds it. So other molecules that are polar will dissolve in water because water's a polar solution. Whereas molecules that are nonpolar, they won't dissolve, and instead they'll group up together. So oil is nonpolar. You put it into some water, it is hydrophobic or water-fearing, and therefore it all comes together and makes little droplets. You've had oil in water, you've seen it at some point. Yes. Awesome. So think about this, right? Your body's got hydrophobic and hydrophilic fluids moving through it and substances in it. Like any fats that you've got stored in your body are in the form of oils. So if you are exposed to a poison that is also hydrophobic, it can get into those oils, the regions of your body that are hydrophobic, and just kind of sit there, all right? And remain in your body for a long time, because it won't flush out with water. Your blood is an aqueous solution, so chemicals that are hydrophilic, they can go through your bloodstream, they can be sort of dealt with differently in your body, and they can be recovered and moved through. You just drink enough water, you'll flush it all out, you know, that sort of thing. But it really all comes down to, is the poison hydrophobic or is the poison hydrophilic? And that's going to dictate a ton about the poison's fate as it moves through an ecosystem, because it's moving through organisms, right? If there's an environmental contaminant and then it gets sucked up into a plant and an animal eats the plant, and then the animal excretes waste into some water, and if that poison was hydrophilic, it just moved all the way through that whole system. All right, it's not going to reach, maybe it doesn't even reach a dangerous level inside of the organism that ate the plant. But if it's hydrophobic and the organism consumed it, then it can accrue and build up in the organism and get bioaccumulated and reach levels where it becomes really, really harmful. Just something to think about, okay? Just something to think about. And then your body does have ways of dealing with hydrophobic substances, okay? And we have ways in nature of dealing with hydrophobic substances. It's called soap. That's what soap does, is it takes the hydrophobic sheet of oil that's covering all of our bodies, and it sort of makes the hydrophobic molecules become soluble in water, all right? So a real actual solution to the accumulation of hydrophobic contaminants in, say, a soil is to pour soapy water on it. You call it something different, and there's a company willing to sell you soap for 100 times the market value of soap, but they put a different label and a different name on it, and they sell it as a soil surfactant, and it sounds different, okay? But it's the same chemical. Anyway. That's enough of that. Stuff moves through the environment. Hey, what are the sources of these poisons? All right. Source could be industry, agriculture, domestic sources, etc. You got all these poisons, you pour them down the sink, okay, and they go into the water system. And what happens there? A lot of things can happen. What happens in soils? A lot of things can happen. What happens in living things? A lot of things can happen. All right? These things can be, essentially, it all boils down to the poison moves through the system, okay, from location to location. Sometimes it's in an organism, sometimes it's in a body of water, sometimes it's in the sky, sometimes it's in the soil. And at each one of these stages, it can break down, or it can leave, or it can accumulate. Those are our three real options: accumulate, break down, or leave the system, or leave that stage, that location within the system, right? So if you look at, for example, the biota right in the beginning, right in the middle of this figure, we've got those three big categories of, hey, metabolic breakdown, store the compound, or excrete it. And really, all of these, like these complicated things in water of hydrolysis, oxidation, microbial degradation, evaporation, sedimentation, this is just those same concepts, but of it either stays there, it moves out of there, or it's broken down. Cool. There's just different ways that these things can be broken down. How do we get exposed to these things? Okay. Some cosmetics and household accidents, or accidental exposure to chemicals. I mean, boy, I think that's a crazy glue container, and it sure does look delicious. You know, it's in that cute little packaging. You drink water, you put some drugs in your body, you get exposed in your job, okay? You eat some food, you breathe some air, all right? You can get exposed to poisons in the environment, all right? Or to other compounds that are harmful to the organism. Because remember, poisons, they're only poisons if they are harmful through their chemical action, all right? Awesome. So we've talked about exposure, movement through systems. Now we can talk about hydrophobic and hydrophilic molecules. Now we can talk about bioaccumulation, okay? So bioaccumulation is when an organism gets exposed to a poison, a toxin, whatever it is, okay? And here it is again, selective absorption and storage of toxins. I'm going to keep changing it. I hate that so much. It's not the real word. Okay. All right. Bioaccumulation. All right. You keep getting exposed to something in small quantities and it gets stuck in your body. Maybe it's hydrophilic as one example. And your body just keeps, sequestering it. Some plants, if they're exposed to, say, heavy metal contamination, they'll just take that heavy metal and, oh my gosh, what's the word? They bind it up. They chelate it. So they bind it all up, make it inactive, and store it in a certain portion of the plant, or store it in a certain portion of individual cells. So like, let's say you're growing a bunch of lettuce in a lead-contaminated field. Well, that lettuce is growing up, and it's got a constant sort of low-level exposure to lead. But as it sucks up water that has lead in it, that lead never leaves, so it starts to bioaccumulate. So it reaches a higher concentration inside of the organism than in the ambient environment. So bioaccumulation is on the individual organism level. Like, this is a biomagnification example, so let's not worry about that too much. But like the lettuce, let's just go with the lettuce, because that's a real world example. Lettuce is used for bioremediation of lead contaminated soils, because it sort of extracts lead that was in the soil into the plant. You cut it, you throw it somewhere, and then you just don't think about it, all right? Because the lead's gone now, right? Next, we got biomagnification. So biomagnification occurs in a food chain, essentially, where you've got these these species that are low on the trophic levels, right here, these small insects, and they're being exposed to DDT, okay, in the environment. And the DDT isn't degraded or excreted at a faster rate than it is taken into the insects, so it bioaccumulates. And then you've got these fish, and these fish are consuming the small insects. And once again, they're eating a lot of these insects in their life, so they're constantly taking in DDT. It's not degrading or being secreted rapidly enough, so it bioaccumulates in the fish. And then the next level, the gulls. They're eating the fish. Now the fish had higher levels of DDT than the small insects, which had higher levels of DDT than the bottom mud, all right? And therefore, they're getting exposed to a higher level. They eat multiple fish in their lives, and so the concentration of DDT gets even higher in the gulls. So this figure right here is an example of biomagnification, but biomagnification sort of requires bioaccumulation, because if bioaccumulation is not happening, then the compound doesn't build up. Does that sort of make sense? Yes, please.
So is it like bioaccumulation is specific to an organism, whereas biomagnification is like what is caused when lots of organisms bioaccumulate?
Yeah, exactly. I'm just going to repeat it for the recording. Let me know if I got your point there. Bioaccumulation is for the specific individual organism, whereas biomagnification is, I'm going to rephrase a little bit, a food chain where all of the members are bioaccumulating. That cool? Awesome, great. That's good clarification. So these are two other pieces of terminology that are misused sometimes, And it really kind of matters because in a complicated trophic structure, because I've showed you before, like those complicated wibs that have the different actual numbers associated with them for the feeding rates and all that. If one member of that complicated food web is the bioaccumulator, and that's leading to biomagnification, well, maybe that's a target for control. You need to look at that individual member of the food web and think about, an extreme example would be getting rid of it, but more realistic solution would be controlling that individual's exposure, right? That species exposure, so that it never has the chance to bioaccumulate. But if you don't know the difference between the terms, you don't have the vocabulary for, interacting with a complicated concept like that. Anyways, persistence, a lot of text here. I got super caught up in this topic, and you'll see when we hit future slides that I get a little bit carried away, but that's okay, it's important stuff. All right, it's stuff that I think this course is really supposed to inform you about. I already talked about this a little bit. Persistence. Some things are more persistent than others, okay? Some compounds degrade naturally in the environment. So you want, for example, pesticides, which remember is a general term that includes herbicides and insecticides and fungicides and all of these different things. Okay, a pesticide kills pests, kills living things that we've deemed pests. All right. A good pesticide should have low persistence, right? Because you want it to degrade once you've sprayed it and put it into the environment. You want it to kill the thing, and then you're done with it, okay? Now, oftentimes glyphosate, which is Roundup, the active compound in Roundup plants, is identified as a high persistence environmental contaminant or a high persistence pesticide. But I'm going to make an argument for you in this class that just like how BPA became a focus for regulations and controls, and it actually adjusted people's purchasing habits, pretty drastically. The adjustment to like reusable water bottles. If you were around, 15 years ago or something, you saw the shift as BPA became more of a talking point. People really did start to use reusable water bottles and metal water bottles more. It just became more of a market. Now, the same thing sort of happened with glyphosate, where glyphosate becomes the example of everything bad about pesticides. Glyphosate has huge problems. You've heard about Roundup Ready plants. It's an overutilized pesticide. It's super effective. It works really well. But to use it as an example in every time you're talking about any negative aspect of pesticides sort of ignores all of the other pesticides that are out there. So once again, I'm going to kind of beat up on this textbook, okay, that I keep referencing. They use glyphosate as an example of a high persistence pesticide. But if you actually look at the research about how persistent glyphosate is in the environment, it's a pretty low persistence. It degrades pretty rapidly, all right? So for example, glyphosate has a half-life of as little as 7 days in some soils. So that's really low, honestly, for a pesticide. So if they're like, hey, look, glyphosate's the most widely used herbicide in the world, and it's a persistent organic pollutant. It's like, there are so many others to be thinking about. Anyways, I'm not a Bayer glyphosate Monsanto apologist here. I'm just trying to say that, sometimes focusing on one bad chemical can make things oversimplified. Also, I'm going to do a tiny soapbox. It leads to a lot of really bad research, which then muddies the waters. If you try to find decent work, it took forever to get glyphosate recognized as a carcinogen, and not only because of the companies trying to stop the research, but because everybody wanted to do research to prove that it was carcinogenic. And so many studies ended up redacted and being produced incorrectly, but still getting published because it was a big buzzword. And it's really easy to be like, well, it's not carcinogenic. Look, there were 100 articles last year that were all misrepresenting their data. Like, people just don't like us, I don't know. Whatever. Let's keep moving forward. Persistence. Okay, do we understand the term? Are we comfortable with the term high persistence versus low persistence? How do we measure persistence? Usually as half-lives. Okay, what's the half-life of a compound? How long it takes for half of it to go away. Okay, so it's a pretty reasonable representation of the rate of loss of a contaminant in the environment. Let's say that it contaminates at a constant rate, okay? It's got this many days would be its half-life, okay? So it keeps going halves and halved, right? But really what these tend to look like, these curves, are not linear lines. They tend to look more like this in the environment, where the degradation rate looks like that, okay? And so the half-life is like, well, you hit the half point in the concentration. It's very possible, if this is a 7-day half-life, that the compound could still be present in the environment well beyond 14 days. It's just that it takes 7 days for the concentration to be halved in the environment. Does that sort of make sense? Okay, awesome. So that's how we measure persistence. Here's a bunch of different pesticides, and these are what we call persistent organic pollutants, okay? So they're organic compounds, they are pesticides that are designed to kill certain things, but they persist in the environment pretty aggressively. And these half-lives can be like 90 days, 120 days, you know, long periods of time where they stay in the environment, they're accumulating in organisms, they're bioaccumulating, and they're being biomagnified. Cool? Okay, but how we deal with them and how we regulate the compounds, it does matter if they're persistent or not. You don't want to regulate the use of something as if it was persistent if it's not persistent, because the real issue is acute exposure, getting exposed in big quantities all at once, not a sort of chronic exposure to the compound. And not really, the fear isn't really that the glyphosate's going to enter into the whole food chain. It's probably going to degrade in the soil, but the problem is if you apply a ton of it at once, it gets into runoff, it gets into water supplies. The people working on those crops are all getting exposed in massive amounts. So now you, I hope, see how you want to regulate glyphosate use, you know? But you don't want to regulate it necessarily as if it was a persistent organic pollutant just because it's the big boogeyman of pesticides currently. Okay? All right, things can be bad in different ways, and knowing why they're bad, knowing how they're bad, helps us to mitigate the negative impact. A bit of a soapbox day today, but that's gonna keep happening because we're gonna start talking about people getting hurt by bad actors, you know, as we talk about waste disposal. And then we're gonna talk about inefficiencies in waste disposal and people getting harmed, and then I don't know, then all these companies. We'll talk about Keep America Beautiful, which is gonna be a hoot and a holler. Anybody remember this? Everybody, I barely remember this advertisement campaign. Do you remember this? The crying Native American who's actually an Italian actor who was hired. Yeah, you've heard about this. Yeah, this was still a thing when I was a kid, and this group that produced this ad, it still exists. It's called Keep America Beautiful, and you can see them sponsoring activities like the Great American Cleanup, okay? But who's Keep America Beautiful? Anheuser-Busch, Coca-Cola, AT&T, Home Depot, Dow Chemical, iHeartMedia, Hilton. Thank you, all of them, for taking good care of us. Target's up there, you know? And it's all about controlling pollution through litter cleanup, okay? Without ever focusing on wasteful production, okay? Which is really interesting. So one way to think about Keep America Beautiful is it puts the responsibility on us, the consumer, to pick up the garbage and recycle the plastic without ever looking at the companies that are generating all of this plastic waste that the recycling infrastructure is not actually capable of dealing with. So I'm gonna rant and rave about that, and I hope that it's not too obnoxious, and I hope we have a nice time in the class. For the rest of next week, it's Friday, it's the weekend. Don't forget I went a minute over, I'm so sorry. Don't forget to do your discussion. Talk to me about your annotated bibliography. Have a good day, everybody. Goodbye, I'm ending the recording.